Hello, everyone, and welcome to today's webinar, Benefits of Real-Time PCR Multiplexing. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. You will also notice a survey window at the left of your screen. Please feel free to answer the survey questions. We welcome your feedback. I'd like to now welcome today's speaker, Dr. Jon Pfeifer. For a complete biography of today's speaker, please visit the Presenter tab at the top of your screen. Welcome, Dr. Pfeifer. You may now begin your presentation.
Thank you for that introduction. My name is Jon Pfeifer. I'm a senior field application scientist for real-time PCR with Thermo Fisher. So welcome. Thank you for being present for this webinar. So let's get started. The topic today is multiplexing in real-time PCR experiments. Here are some of the key topics. I'll provide a brief introduction to real-time PCR. I'll talk about the benefits, various multiplex scenarios, and multiplex verification. So I'll start out with an introduction to real-time PCR, which is a technology that allows amplification to be detected automatically each cycle. Real-time PCR can be used for many different purposes or applications. There's quite a list here, as you see. I don't intend to go through that whole list. Let's go ahead and talk about the fundamental components. For real-time PCR, we need instrumentation, and that instrument will come with software. We need reagents, assays, samples, and some type of reaction vessel. In real-time PCR, we're collecting fluorescent signals throughout the process, but for analysis, we need to select one special data point from each amplification plot. That data point is usually referred to as a CT, although some software may call it a CQ, but it's the same idea. So once we have those CT values, we can then deduce the initial target quantities in the PCR reaction. The traditional method for calculating CT values involves setting a baseline and threshold value. But just to let you know, there are some real-time PCR programs that will offer an alternative method known as relative threshold. I'd like to define some terms. A target is a gene that the researcher wishes to quantify in samples. Typically, real-time PCR is used with biological type samples, so we need to know sample mass in order to calculate target concentration. And typically, researchers will use a gene to measure that sample mass and then normalize the target to sample mass. So we can call that gene a normalizer. There are other terms too, a control gene, endogenous control, some people call it a housekeeping gene. In our real-time PCR software, normalizer is often referred to as an endogenous control. Defining an assay. An assay is a combination of oligonucleotides used to amplify and detect a specific genetic sequence. In real-time PCR, assays usually consist of either two primers and a TaqMan probe, or two primers in the case of cybergreen-based assays. In this presentation, you're going to hear the suffix plex used quite a bit. It's used to categorize real-time PCR experiments based on the number of assays per well. Singleplex describes an experiment in which there is only one assay in each well. In the example shown on this slide, one target assay was colored blue and the normalizer assay was colored green. And note they're in separate wells. A multiplex describes a real-time PCR experiment in which there are two or more assays present in every well. So now we have the target, blue, the normalizer, green, present in every well. Some researchers may be familiar with single nucleotide polymorphisms, and we have assays for those polymorphisms, and they involve two PCR primers and two Taqman probes. Now, yes, the assay has two probes, but that doesn't mean it's a multiplex because it only has one pair of primers. So we consider this not being a multiplex. Qualitative multiplexing means we're going to detect two or more gene products from a single PCR reaction, but we're not trying to necessarily quantify them. An example of this is the TaqPath COVID-19 combo kit, which is used to detect the presence or absence of the SARS-CoV-2 virus in patient samples. There's also quantitative multiplexing, which is not only are we detecting two or more gene targets from the single PCR reaction, but we're also going to try to quantify them to determine the original concentration of each gene in the sample. The simplest multiplex is a duplex, where we have two assays, typically one target and one normalizer. Engineering a duplex is relatively easy and the success rate is high. We get a little more complicated with a triplex. This is a multiplex consisting of three assays, typically two targets and the normalizer assay. Engineering a triplex is more complicated than a duplex, and there's a growing risk of encountering problems. So this leads to the question of deciding to multiplex. Now, all real-time PCR experiments can be performed in singleplex, but multiplexing can be advantageous. So the researcher should weigh the benefits of multiplexing versus the effort to engineer it. Diagnostic assays may be good candidates for multiplexing more than two assays, as many samples are expected to run over the lifetime of the assay. So let's talk about some of these benefits. There are many benefits to multiplexing: improved quality control, throughput, reagent consumption, sample consumption, and improved precision. And I'll go through each of these multiplexing benefits. First, we have improved quality control. So a control assay of some sort is used to assess whether an acceptable amount of sample was present for the target assay. So if you imagine doing a singleplex assay, it's always possible that the amount of sample delivered to the target well could be different from that to the control well. So there's a bit of a question that if there's a problem with how much was in the control well, does that mean that the target had enough sample, things of that nature? But in multiplex, that problem is mitigated because the sample's being placed into a well that has both the target and the control. So that improves quality control. Also, the next benefits I'll be talking about, increased throughput, reduced reagent and sample consumption, have to do with the number of targets that are in the multiplex. The more targets you have, the greater the benefit. One of the points I want to bring out, though, is what happens if you have more targets that are in the multiplex. Let's imagine, first of all, we'll start out with a singleplex experiment. We've got one target, I'll call it T1, and the normalizer, which I'll designate with an N. In singleplex, a 96 well plate is illustrated on the left. I could theoretically run up to 48 samples per plate. But if I duplexed that, I could now run up to 96 samples per plate. So I've doubled my throughput, which is, I think, significant. But what happens if I have more assays than the multiplex? So let's say now I have three assays, two targets, I'll call it T1, T2, and the normalizer N again. In singleplex, I could run up to 32 samples per plate, 96 well plate. If I were going to run this as a duplex, I'd have to have two of them. So I can now run up to 48 samples per plate. So my benefit, instead of a twofold in terms of reduced sample consumption, etc., has now gone to only 1.5-fold. So what we can see from this is that as the number of assays that you're using in your experiment exceeds the multiplex number, the throughput and consumption benefits diminish, as shown in this graph. So to take full advantage of multiplexing benefits, it's best if the multiplex incorporates all the targets and control assays. But as the number of targets increases in the experiment, you may hit a point where you may want to consider an array instead. So there is a limit to the maximum number of assays that can be multiplexed in real-time PCR. And when you have a larger number of targets, an array might be a good idea. So an array is a PCR vessel in which the assays are dried down in specific well positions. I'm showing 4 kinds of arrays in this slide. We have 96 well Tachman array plates. We have 3 to 4 well array plates, Tachman array cards, which have 384 microwells. And then we have the amazing open array with 3,072 wells, which is pretty amazing. So I'll use a 96 well array as my example. So let's say I have a choice. I could run the 96 well array singleplex, or I could duplex all of those assays. Well, first of all, the benefit is instead of being able to measure 95 targets, I could now measure 96 targets. That benefit is minuscule, but also there's an issue because that would mean having 96 duplexes. I'd have to verify the performance of 96 duplexes, which may be rather cumbersome. But on the plus side, with an array that, assuming let's say singleplex, that you get a special precision benefit if the master mix that you're using contains a passive reference dye, such as rocks. So in other words, in hand pipetted, let's say, individual assays, you're not using an array. Rocks normally wouldn't give you a precision benefit when it comes to pipetting the sample. But when it comes to arrays, it does. So throughput and consumption benefits from multiplexing increase in direct relation to the number of assays in the multiplex. However, development effort increases, I could say, geometrically with the number of assays in the multiplex. So again, this is where I'm urging researchers thinking about multiplexing more than 2 assays per well, consider the practicality. In other words, how fast could you get the results in singleplex versus the time required to develop the multiplex? So another benefit of multiplexing is precision. So if we imagine a multiplex where both the target and normalizer are present in the same well, that means when you deliver sample to that well, it's the exact same amount of sample for both the target and the normalizer assay, which differs. In a singleplex, the pipetting of the sample for the target and normalizer assays are independent of each other. So you can have precision issues there. The good news here is that if you do a multiplex in our real-time PCR systems, our software will recognize the multiplex immediately and perform well-based normalization. So in other words, the target in, let's say, well A1, that data is normalized to the normalizer in well A1. Since it's the exact same amount of sample, whatever error there was in delivering sample to that well is the same in the numerator as it is in the denominator. The error cancels out. So you're getting better precision. I'm showing a real-life customer example to illustrate the benefit of the multiplex for precision. So this was a multiplex experiment, and what I was able to do is calculate what the precision would have been if it had been a singleplex experiment. And those are the blue bars that you see there. And then the actual precision was the multiplex in red. And you can see that in most samples, the precision of the pipetting was quite good. So we get similar precision with the singleplex and multiplex. But there was one sample on the right where there was a problem of some sort, but the multiplex precision benefit really, really helped. Another point is if you have an assay that is not robust, as shown in this example, we can see in this amplification plot, as we go from left to right in the amplifications, that the signal is getting weaker and weaker and weaker. So there's some kind of problem with this particular assay. It may not be a good candidate for multiplexing. You want a nice, healthy, robust assays when you're going to engineer a multiplex. Our concern, which is pretty rare, but it's something to be aware of, is extreme abundance limitations. The real-time PCR has an amazing dynamic range potential. We can literally have billions of starting DNA copies of the target, and the exponential phase remains consistent. But the dynamic range is not infinite. It is conceivable that one could add so much initial target that we get a CT very, very low, maybe less than 3, perhaps. And when that happens, the baseline may be so small, it may be difficult to set a good baseline for baseline subtraction. So there is a limit. I rarely see that limit, but I have seen it on a few occasions. Now, how this relates to multiplexing is, let's say one decides to multiplex 2 assays. One assay is for a gene that's very abundant, maybe like ribosomal, for example, and another one is for a very low abundant target. So if you need to add a large amount of sample in order to see the low abundant target, it's possible, maybe, that the highly abundant target may max out PCR. So that might not be a good idea to multiplex those two. Another point to be aware of is adverse primer or probe interactions. So in a duplex, we only have, in a singleplex, we only have 2 primers. We start adding more primers in a multiplex. So there's a greater risk that those primers could interact with each other, form primer dimers, things like that. And that could have an adverse impact on the assay. Now, there are algorithms to predict whether these adverse interactions might occur, but I think it's wise to always test the multiplex using wet chemistry to be sure. If the intent is to multiplex our TaqMan assays, then please contact our Thermo Fisher technical support. We have a bioinformatics team that can check for potential adverse primer-probe interactions because with the TaqMan assays, for obvious reasons, we don't provide those primer-probe sequences, but we can still do the check for you. Let's say you've decided, yes, I'm going to multiplex. What components do we need for successful multiplexing? Chemistry, instrumentation, dyes, and assay formulation. And I'll talk about each of these. First, the chemistry. So probe-based chemistries like TaqMan are recommended for multiplexing because they're easier to design than compared to DNA binding dye assays. It's easier to analyze because each gene has its own unique reporter dye, and the probes provide specificity protection, which becomes increasingly important with the more assays in the multiplex. It is possible. I have seen duplex cybergreen assays, but the design is clearly more difficult. Multiplexing is more limited, and there's more concerns about problems that may occur. If one is working with an RNA application like gene expression, we have to reverse transcribe the RNA, turn that RNA into DNA referred to as cDNA. Reverse transcription chemistries can be divided into 2 categories called the 1-step and the 2-step. In the 1-step, it's a single chemistry. The researcher places the RNA in the chemistry, reverse transcription happens, and immediately afterwards PCR happens without user intervention. In the 2-step chemistry, the researcher prepares a reverse transcription mix initially, making cDNA. The cDNA is then transferred to the PCR reaction. In the 1-step chemistry, the reverse PCR primer is used for cDNA synthesis. Because of that, the reverse PCR primer concentration can have an impact on reverse transcription efficiency. So this can mean that engineering a 1-step multiplex may be a little more complicated compared to that of a 2-step, because the forward and reverse PCR primers may need to be at different concentrations for optimal performance. You can see in this example that parameter titrations were performed for multiplex using the one-step chemistry, and there's a relatively small region, we're calling it the plateau, where the parameter concentrations are optimal. But that's not to say you can't multiplex with a one-step. You certainly can. They can be successful. This is an example shown here of a triplex using the TACPath one-step master mix. Then we have the two-step. Typically in the two-step chemistry, random primers are used to prime for cDNA synthesis. And by the way, it's okay if there's some oligo-DT as well, but we're just recommending against using pure oligo-DT for priming in the two-step chemistry. So in that case, with the two-step chemistry, as is often used with gene expression experiments, the concentration of the forward and reverse PCR primer can be the same because they're not involved in priming cDNA synthesis. So in this example, where a two-step chemistry was optimized for multiplexing, you can see a much larger plateau region for optimal primers. Another question is, how many dyes are you going to need for the multiplex? And here are 3 rules to follow. 1 reporter dye per assay, or you could say gene. If there are any quenchers that are dyes, we need to count those, but non-fluorescing quenchers, you don't have to worry about those. And number 3, if there's a passive reference dye, which is always optional, but could be very useful. In fact, pretty much is always useful. So let's take an example to illustrate. Let's say that I want to do a duplex. So I've got my target assay labeled with FAM. The probe has a FAM reporter. And this particular probe is an MGB type of Tachman probe. Now, MGB probes have a non-fluorescing quencher, NFQ, on the 3' end. So we don't have to count that quencher as a dye. It's not a dye. So far we have 1 dye. The normalizer, let's say, has Vic as the reporter, and the quencher this time is Tamra. Now, Tamra is a dye. So far we're up to 3 dyes, FAM, Vic, and Tamra. And let's say I'm going to use a passive reference dye ROX. That's a 4th dye. So it's a duplex, but I have 4 dyes in my chemistry. Now, something that I have to mention in regards to this is we have many different real-time PCR instrument models, as you can see from this list, and their excitation types can be divided into 2 categories, multi-wavelength excitation and blue excitation. Now, you might wonder, why does this matter? So red dyes are not efficiently excited by blue light. However, even though ROX, which is our passive reference dye in our master mixes, it is red, but the formulation of it is such that it is efficiently excited by blue light. So to use an example to illustrate that, let's say I have a step 1 plus, which is a blue excitation system. So it has 4 filters. So I could handle 3 reporter dyes, or 3 probes, one could say, plus ROX as the passive reference. But I could not use, it probably wouldn't be a good idea, to have a red reporter, because blue light doesn't excite red reporters very effectively. Multi-wavelength excitation means that the excitation source is going to have broad frequencies, and those are divided up into filters. And so the filters can be paired, that is, excitation filters can be paired with emission filters to provide maximum excitation. So therefore we can use a wider range of red dyes. So then logically the next question is, well, which dyes do I pick? Here's some general rules to follow. The dyes must be commercially available for oligo labeling. The dyes must produce sufficient signal in the instrument you intend to use, and dye combinations in the same well must be accurately resolved by the real-time PCR system. Now, you don't have to follow the pattern that we follow, but we follow a pattern when it comes to dye selection. We start with the shorter wavelengths and then move gradually to the longer wavelengths. So the first dye we typically use for Techman probes is FAM. That's a green emitter, shorter wavelength. And then we can, if need to, multiplex, we go with VIC as our second dye. If we need to go to a third dye, Abby, and we have a fourth one, June. And one of the rules is not to put in the same reaction dyes with the same color. So for example, for orange, we have Ned, Abby, and Tamara. So Ned and Abby we can use as reporters. Tamara, we use those as a quencher in some cases. So these dyes, like Abby, Ned, Tamara, those should not be together in the same chemistry. And if one intends to use the orange channel for multiplex, it's best to avoid Tamara quenched probes. That way it frees up the orange channel. Also, I had said earlier that ROX is a red dye, so is June. So we don't want to have both June and ROX in the same chemistry. If you do want to use June, we have an alternative passive reference dye called Mustang Purple. Mustang Purple can be used in multi-wavelength excitation real-time instruments with at least 5 filters. And we have master mixes that contain Mustang Purple. I'm showing 2 examples here called the TACpath One-Step Multiplex Master Mix and the TACpath ProAmp Multiplex Master Mix. So those who want to go even beyond 4 dyes, which is significant, I would say, we do have what are called far red dyes, and there's 2 called Psi 5 and Psi 5.5. Probes with these far red dyes can be ordered from Specialty Oligos, which is a part of Thermo Fisher. And by the way, Mustang Purple should not be used if you have a Psi 5 probe, because they would emit through the same filter. I think this is a fairly obvious statement, but the more dyes you have in the multiplex, it's more important to maintain current instrument calibration. That's beneficial regardless, I would say, but even more so in a multiplex assay. So I want to show an example of a calibration problem. So the symptom of a calibration problem is when the amplification of one assay causes a response in another assay that's not amplifying. And so in this example, VIC, which is colored green, is amplifying. This is causing a response in both ROX. If you can see, there's a little increase in ROX there, and there's a dip in the FAM baseline. Turns out that the reason for this calibration problem was that the VIC supposed dye was not actually VIC. It was a different dye called hex. So this is telling us that the spectral shape of VIC is not an adequate mimic to that of hex. It's also, I think, useful to point out that we offer 2 different Techman probe types called MGB and you could say non-MGB. Techman MGB probes are shorter compared to non-MGB probes, and they have enhanced specificity. In other words, we could say that mismatches in the probe binding site have a greater impact. The probes are more sensitive to those mismatches. MGB probes, by the way, have a non-fluorescing quencher on the 3' end. Then we have non-MGB probes, and we offer them with different quenchers. There's the traditional Tamara dye as the quencher on the 3' end, and there's a QSY non-fluorescing type of quencher as another option. Non-MGB probes are longer than MGB probes. They're a little bit less sensitive to mismatches in the probe binding site. So by choosing MGB or non-MGB, you can, in a sense, tailor the specificity of the assay a bit. If you go to our website and look at probe dye selection, you can see they are going to be a little bit different whether you choose an MGB or a non-MGB probe. Now, each Applied Biosystems brand real-time PCR instrument has a list of standard dyes that it will be calibrated for. If one wishes to use dyes that are not part of the standard dye list, we call those custom dyes. And so we recommend that a custom dye calibration be performed, which I would argue is not that difficult. You have the dye in a plate, and that plate is then read in the instrument. Another important concept that those who are trying to engineer multiplex should be aware of is polymerase saturation. Polymerase saturation occurs when the amplicon concentration reaches a point where there's not enough Taq DNA polymerase to support geometric or exponential amplification. So when designing a multiplex, it's a good idea to think about the potential for primer saturation, and if it looks like that is a possibility, to take steps to prevent it. So I'm going to illustrate polymerase saturation with a hypothetical example. So let's suppose that I have a duplex. I have an assay for a gene which I've colored green, and that gene is more abundant, let's say, in my samples. Then I have a second assay which I've colored blue, and this is for a lower abundance gene. So when I run the experiment, I first hit start. Initially, both the green and the blue genes are in the exponential or geometric phases. And that's good, that we want to maintain that relationship between initial target quantity and geometric amplification. So far, so good. Then the more abundant gene signal will emerge from the baseline first, and it's still in the geometric phase. We still have good data here, but we're going to wait on the lower abundant gene for its signal to emerge. So far, so good. Problem though is, what if the green gene assay has a high primer concentration? For example, our universal primer concentration is 900 nanomolar. That would allow the green gene to continue to amplify to the point where it saturates the polymerase. So now the assay transitions from geometric to linear phase. The problem that causes is in a multiplex, when one assay goes from geometric to linear because of Taq saturation, the other assay has to do the same thing at the exact same time. And if that happens before the lower abundant assay signal emerges, then you're going to have a problem. You're not going to detect the geometric phase. You might see linear phase perhaps, or you might see nothing at all. So like I said, if you did see linear phase data, it will be inaccurate, and we shouldn't use that for quantification. So we can call this a failed multiplex. The solution to prevent polymerase saturation is primer limitation. In other words, the assay has lower primer concentration, so that the reason the assay for the more abundant gene transitions from geometric to linear phase is not because the polymerase was saturated, instead because primer ran out. This means that there'll be plenty of polymerase for the downstream assay to amplify in the geometric phase and detect that geometric data. One could do a manual primer limitation. That would involve a primer titration experiment. The goal is you want enough primer to detect the geometric phase, yes, but low enough that you prevent saturation. So I want to show a real-life customer example of a failed primer limitation experiment. So it's a duplex. We have, I'll call it gene 1 and gene 2. There was a manual primer titration experiment that was done. In the initial tests, there was concerns that the data was not accurate. That is, the gene 2 data points were not accurate. And we can see why in this amplification plot. Gene 1 is producing a very robust geometric phase. That looks fine. But notice gene 2, those lines are not parallel with gene 1. What we're seeing is linear phase data with gene 2. So that explains why it's inaccurate. Also note that the plateau for gene 1 is greater than 1, which suggests that a relatively high primer concentration was used for gene 1. But the same assay, there were some samples that gave accurate results, and in those cases, that gene 2, the CT values, were similar to those of gene 1. So in other words, they have similar abundances. So this is illustrating that when you have two genes and there are similar abundances, they're going through the geometric phase pretty much at the same time. So polymerase saturation is not an issue in those cases. But obviously, in other cases, other samples, gene 1 was significantly more abundant than gene 2, so primer limitation was important. Another assay done by the same lab, this time it was gene 3 paired up with gene 2, and we can see that gene 2 now is giving parallel geometric phases compared to gene 3, which is what we would hope for. Notice too, the plateau phase for gene 3 is significantly below 1. So that would suggest a much lower primer concentration was used for gene 3, and I think this is an example of a successful primer limitation. But actually, you don't have to do a primer titration experiment to identify primer limitation. We offer assays that are primer limited right off the website. One example of that is that we have a category of assays called endogenous controls. So these are genes commonly used to normalize in gene expression experiments for human, mouse, and rat. And we offer them in different formats. One format is VIC, and VIC format is automatically primer limited. Also, just parenthetically, I like to get the endogenous controls large scale, and you can tell that because it has the E suffix in the part number. Then you're getting enough for thousands of reactions. The cost per reaction goes down to pennies. So I think it's a really economical way to be able to multiplex with a normalizer in gene expression experiments. But in addition, if you go to our website and look for TaqMan gene expression assays, you can always choose to have the assays VIC primer limited. So when you find a TaqMan gene expression assay on our website, the default, it's called size/die, is FAM-MGB. So it's a FAM probe. It doesn't say it, but it uses universal primer concentration. But one of the choices is VIC MGB PL. PL means primer limited. Also, for those who might be using our custom TaqMan assay design tool, you can also choose VIC PL. And if you're not using any of those options, that's fine too. We have specialty oligos that can synthesize any assay in a primer limited format. But a key question then is, which assays should be primer limited? And to help make those decisions, I have what I call multiplex scenarios. So I'll start with what I guess arbitrarily call scenario number 1, and that is one gene will always be more abundant than the other gene in all samples. A good example of that is gene expression, where the normalizer gene is very abundant, maybe like ribosomal, GAPDH, things like that. And so we would never expect the target to be more abundant than the normalizer. So in that case, it's only the normalizer assay that needs to be primer limited. The target assay does not. An example of that is shown here. So we have a target assay in blue, and you can see it's plateauing to a much higher point compared to the normalizer or endogenous control, which is kind of a greenish color. And the plateau for that, the endogenous control, is much lower. So obviously that one was primer limited, and it was very successful because you can see we have very healthy, nice geometric amplification of the target assay. I'm defining scenario number 2 is a duplex in which the two genes have approximately equal abundance in all samples. I would probably never see this in a gene expression experiment, so this is usually observed in a genomic DNA application, such as copy number variation. In theory, primer titration would not be needed to prevent Taq polymerase saturation if the two genes are always very similar in their abundance. Duplex scenario 3 is when it could be either way. That is, One gene could be more abundant in some samples, and the other gene could be more abundant in other samples. So in that case, the thing to do would be to primer limit both assays. Step number 4 is where there are multiple assays, yes, but only one is expected to amplify in any particular sample. So in theory, primer limitation would not be needed unless you have a normalizer assay that's included in the multiplex and you're concerned that strong target amplification may suppress amplification of the normalizer. And an example of an internal control is a product we have called the internal positive control, or IPC for short. So this is a plasmid, and it contains a region that is, as a sequence, completely unrelated to anything found in nature. A positive control like this, or internal positive control, becomes very valuable in a qualitative test or experiment. It's used when the target fails to amplify, because then you want to have some assurances that the sample was fine, there was no inhibitors, things like that. So if both the target and the IPC fail to amplify, you know there's something wrong with that particular sample. But if the target failed to amplify but the IPC amplified normally, then you can say that that's most likely a valid target negative result. Now we come to multiplex verification. So my recommendation is, before using the multiplex to produce data, to verify its performance first. And individual assays will be needed for the verification process so we can compare performance between the singleplex and the multiplex. But once verification has been completed, the assays can be formatted all together into a single tube, if you wish, by using our specialty oligos. Multiplex verification involves mixed standard curves. That is, one target will remain at a fixed concentration while the other target has varying concentration. That was like a standard curve. But you're mixing the two together in various ratios that are those ratios expected in real samples. And we're going to compare the mixed standard curve performance in both singleplex and multiplex. And hopefully we don't see a difference. If, on the other hand, in the multiplex, we see that, let's say, if one gene is far more abundant than the other one, and we're starting to see the CT values increase as a result, then we know we have a problem here. We have amplification suppression. Perhaps it's the result of polymerase saturation, perhaps adverse primer interactions. If that's the case, adverse primer interactions, you may need to replace the offending assay. So once all the primer pair options have been tested, if it is a duplex, then you're good to go there. But if it's, let's say, a triplex, then I'm recommending do a multiplex verification where you're now testing that triplex compared to singleplex results with a representative group of samples. I want to make sure that the results are the same within experimental error. And you can see that this verification process gets increasingly important as and also complicated as the number of assays in the multiplex gets larger. So for example, if one was doing a quadruplex, you see all the different possible duplex combinations. It has certainly grown, and there's the sample testing. So again, the point being that the more assays in the multiplex, the verification gets more complicated. So when considering whether to multiplex more than 2 genes per well, I suggest weighing the benefits versus the verification effort. Now, if the plan is to run many samples over the lifetime of multiplex, that development effort is likely to pay off. So that concludes the presentation. Thank you for participating. And now we have time for questions.
Thank you, Dr. Pfeifer, for that informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you'd like to ask, please do so now. Just type them into the Ask a Question box and click Submit. We will answer as many of your questions as we can. So, Dr. Pfeifer, let's begin with this first question. We have a lot of questions already coming in. If I perform a custom dye calibration, how do I know if it worked?
Thank you, Christy. So there are two layers, one could say, of working when it comes to custom dye calibration. So first of all, different real-time PCR instruments will have what we call standard dyes that the instrument is calibrated for routinely. But if it's a dye that's not part of that standard dye list, we call that a custom dye. So first of all, we have the custom dye calibration process. And for that to be successful, all we need is a reasonable amount of dye signal. And that's guidance is provided in the user bulletin, maintenance guide, things like that. That's usually not an issue. What's more important, I think, by way of working, is does the spectral signature of the pure dye that was used in the custom dye calibration process, is that spectral signature accurate for the dye that's on the TaqMan probe? So I think that's a more important question. One way to answer that question is to amplify that assay with this new custom dye. Singleplex is fine. With a chemistry that contains passive reference dye, most commonly rocks, and see if, as a result of amplification, do we get a response in rocks. So if the custom dye calibration was accurate to the dye that's on the probe, we should not see a response. And if it's not accurate, then you may see a response. So then that means that the custom dye that was used in the calibration versus the dye that's actually on the probe, those spectral signatures are not a good match. And so that's going to be an issue that needs to be addressed.
Thank you, Dr. Pfeifer. Our next question, what is CQ confidence and AMP score?
Okay, great question. So CQ confidence, so many of our real-time PCR software programs produce what might be called quality control parameters. There's two of them listed here are CQ confidence and AMP score. CQ confidence is based on the shape of the amplification plot. So in other words, it is the confidence that the shape of the amplification that you're seeing is consistent with a true or ideal, one could say, amplification plot shape. AMP score is a different value. It's based on signal intensity. So for example, if there was some kind of distortion or something that happened, one would think that the signal, say if the signal bounced up a little bit, that it wouldn't be a very strong signal. So it'd have a low AMP score. But if it's true amplification, you should see a strong signal coming up out of the baseline, and then that would give you an indication that, yeah, this is more likely a true amplification rather than a distortion. Specific issue, I would say.
And Dr. Pfeifer, it looks like we have time for just a few more questions. Could you please speak about the CT value?
Oh, okay. Well, that's kind of a general question. Okay. So CT, this is the original terminology, threshold cycle, or CT for short. So the first, and still very popular, most popular way to collect data points from our real-time PCR data. In other words, with real-time PCR data, we're usually going 40 cycles. And presumably, if the target was there, you would see amplification emerging at some point. And so we want to select one special data point from each amplification plot. And that special point is often referred to as a CT. We do have some software where it's referred to as a CQ, but it's the same idea. So where the CT gets its name from is cycle, or threshold cycle, or cycle threshold, how you want to say that. So there's a fixed fluorescent value that's selected called the threshold. And where and generally the threshold would intersect the exponential phase data. And so where you get that intersection, that corresponds to the CT. So the CT has an inverse relationship to the number of molecules of the target you started with. So in other words, the smaller the number of target molecules you had in the reaction, it's going to take longer to amplify to the point that you now start to see signal emerge from the baseline. And conversely, if you have a large number of target molecules, it will take fewer cycles. The CT will be smaller because it took fewer cycles to generate that level of signal. So CT values can either be used qualitatively, like we do in our COVID assay. If you see a CT, great. Well, not great, but you've detected the COVID virus. And if you don't see the CT, COVID virus is not there. And I'm speaking very simplistically for this particular application. There's more to it than that, but if you're going to quantify, then what we need to do is transform those CTs into initial target quantities. And the best way to do that is by using a method called delta-delta CT. So that's my little spiel. Of course, I can go a lot more detail about it, but that'd be the basics of it.
Thank you, Dr. Pfeifer, for this excellent Q&A session. Do you have any final comments for our audience?
So thank you for attending today's presentation. We here at Thermo Fisher are here to help you. So please reach out to your local Thermo Fisher support person. It could be sales rep, depending on your needs, of course, sales representative, your FAS, Field Application Scientist like me, tech support, 800 number there. We want to help you. If you're successful, that means science is successful, I would argue. And if we can help you in that process, I think that means we're successful. So please think of us as your partner. We want you to have successful experiments, and we can help guide you. So again, thank you for participating in this presentation.
And thank you again, Dr. Pfeifer, for your time today, your vast knowledge, and your important research. I do want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I do want to thank the audience for joining us today and for their interesting questions. Any questions submitted during the on-demand period, they will be addressed by Dr. Pfeifer and his team via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and have a great day.